very pleased to welcome Mike Halassa for our brain connectivity workshop. I don't know, Mike, if you know about how we do things, the 15 to 20 minute presentation and the extensive question and answer afterwards. It's a bit of a secret sauce that we've perfected over the last several years. And it's interesting that you, Mike, will talk about control as well. We've just talked about dynamic control theory from Danny Bassett, and in some ways we're looking at control now from a different perspective. Mike Halassa is at MIT and will tell us about his work in the neural basis of cognitive control. Over to you, Mike. Thank you very much.
Yeah, so thanks a lot for the invitation, Olaf and Randy and everybody else. It's really an honor to be here. And, I'm going to talk about very different types of levels of organization than what has been discussed throughout this workshop so far. So I'm going to be focusing a little bit more on the microcircuit scale of organization and sort of network motifs that are defined by single cell profiles. And part of the reason why that is because the preparations that my lab works in is animals that allow us for genetic accessibility and identifying cell types to build these functional circuit profiles that offer us an opportunity to frame our hypotheses about brain function. So that'll become obvious throughout my my talk. So I'm going to talk about thalamocortical interactions in the context of cognition. Before I do that, I'm just going to give you a broad overview of the major goals of my research programs that involve sort of understanding what thalamocortical interactions do in the context of cognition and complex decision making. So that's really the primary focus of what we do. And we use these genetically accessible models that allow us to do precise perturbations in building these wiring diagrams of basic executive functions, things like attention, working memory, integration, those types of things that we think of as the building blocks for higher level cognition. We also work with people who build these large scale computational frameworks for that approximate brain function from the perspective of machine learning, dynamical systems theory, etc. And one part of my research program is also focused on making sense of all of these different elements in understanding things like inference, perturbed inference in psychosis, and what can we do about these types of things once we have a circuit understanding of the process. Okay, so that's sort of like the big picture. And the cognitive process that I'll be focusing about today to introduce to you our work in thalamocortical interactions is goal-directed attention. And to just summarize that process, it's the ability to, based on internal goals, to amplify and suppress incoming inputs. So you can basically imagine that based on what you care about in the world, there are these filters in your brain that you can turn on and off on demand. And the way we imagine this process to work, and this is based on a lot of previous work in humans and non-human primate, we think of this process as separated into two components: an executive component that involves frontal networks, where things like high-level plans and internal goals are represented, because neurons in these areas are tuned to these type of task variables. And by projecting these signals back onto sensory cortical areas, these top-down attentional signals can change the sensitivity of these areas to their incoming input, and that's how we can, amplify the things that we care about, suppress the things that we don't care about. So, in addition to this cortical process, over the last decade or so, a little bit more than a decade, people have realized that there's quite important subcortical contributions to goal-directed attention, particularly this area of the brain that we call the thalamus. And early work from the lab, which is consistent with a lot of work in other people's labs, is that the sensory thalamus, areas like the LGN or the medial geniculate, are a locus for attentional control. So rather than being a passive relay from from the sensors to primary sensory cortical areas. These areas can be modulated by top-down input from the frontal cortex, and we've, along with colleagues, have described detailed pathways that mediate these functional interactions. But most of what I'm going to talk to you about today is not these interesting effects in sensory areas, but rather this really unexpected role for the associative part of the thalamus in attentional control and potentially in cognition more generally. So most of our work right now is focused on non-sensory parts of the thalamus, which in our brains as primates are the biggest parts of the thalamus, areas like the medial dorsal thalamus, for example, which receives most of its inputs, not from the periphery, but from frontal cortex itself, and projects back onto frontal cortex, not so much to relay any type of sensory inputs, but rather to configure cortical representations according to the context at hand. And this idea of cortical configurations by these thalamic control functions is the main focus of my research program right now, and I'm going to tell you about it. Okay, so let me just give you the really the most important computational idea of what this work is about and why, in my mind, at least for me and my research program and my collaborators, this has been a paradigm shift. Okay, so in the thalamus, compared to the cortex, the thalamus is composed of excitatory neurons that are devoid of lateral connectivity, lateral excitatory connectivity. So everything in the thalamus is defined by long-range input-output connectivity patterns. The neurons don't talk to each other. And therefore, the reason why we think that the LGN is a relay is because it has strong driving input from the retina and a strong driving output to primary visual cortex. And consistent with this idea, responses in the retina are center surround, and in the LGN, they're center surround. So from that perspective, the receptive fields are identical to the most part, except for some subtle timing differences. And the real interesting computation arises at the level of primary visual cortex, where neurons start forming these being sensitive to oriented edges, to visual motion, etc., by appropriately sampling from LGN inputs. So by learning these weights and constructing the appropriate sort of input patterns, you can get orientation selectivity in primary visual cortex from these building blocks from the LGN. So this is where the interesting computation is, and the LGN is a relay. Okay. In areas like the medial dorsal thalamus, we think there's a completely different thing going on, both at the level of the inputs and at the level of the outputs. So at the level of the input, we think that there's actually quite a bit of convergence happening, and there's this possibility that you can adjust these weights at the level of the thalamus, so you can generate representations in the thalamus that don't even exist in cortical afferents. And the output, we're starting to recognize, is actually completely different as well. The excitatory component is modulatory, and there's a pretty substantial inhibitory effect on the cortex that's mediated by local circuit interneurons. And I'm going to tell you about that in a little bit more detail throughout the talk. But why would you need a structure like this type of thalamus for an area like the prefrontal cortex or associative cortical area? This is what we think happens. So in an area like the PFC, where cognitive signals can be generated based on very different types of input patterns, like the meaning of words based on very different types of languages, it helps to have a structure that can rapidly compress these input patterns into a low dimensional form, and then use this type of representation, a low dimensional representation, to amplify the task relevant input patterns, suppress the task irrelevant ones, so that the cognitive signals are configured according to the input statistics or the input that are context relevant. The input-output connectivity patterns are adjusted on the fly by having a structure like the cognitive thalamus in the cortex. And the way we know all of these things is by designing behaviors that allow an organism to switch between different types of input patterns generate these invariant cognitive signals and allow us to access the circuits with enough precision to be able to perturb the system and infer these types of functional interactions. And that's what the type of studies that I'm going to be presenting to you today will be. So my outline is twofold. I'm going to tell you, and I'm going to try to limit this to 15 or 20 minutes. But the idea is that I'm going to show you how we did a lot of this work in the mouse, although we're doing these studies in other organisms as well at this point. I can tell you about that if anybody's interested. So we use the mouse to understand how prefrontal substrates of goal-directed attention are generated. And then we use this exact preparation to understand how the thalamus, the cognitive thalamus, reconfigures these task-relevant representations according to the context at hand. Okay, so everything that we were able to do was enabled by being able to control behavior in the mouse in the tradition of behavioral control in non-human primates. So we approached mice as monkeys and asked them to allocate their attention according to a goal-directed memory. And this is how the behavior works. So we train mice on two alternative force choice tasks where they're required to select between a visual stimulus, a light flash that can come to the right or the left of the animal, and an auditory sweep that tells the animal to go right or left. I'm showing two cases of stimulus presentation or target presentation that are in conflict, but they don't have to be in conflict. They can be pseudo-randomized. And in order to get a reward, the animal has to go to the stimulus that's valid for a particular trial. Okay? How does the animal know which of these two stimuli are the valid one? Well, over a period of weeks, we train it to associate a particular cue, and this is completely arbitrary. The mapping is arbitrary across different mice, where we have, this is just an example of a high pass or a low pass burst of noise. And the animal over weeks learns that this means attend to vision, this means attend to audition. At the beginning of each trial, the animal gets randomly one of these two different cues, has to hold it in mind over a brief delay, and then map it onto the appropriate target stimulus. So the only way the animal can solve this task is to encode this cue, turn it into attentional rule, and then use it to map onto the appropriate stimulus. And we've done all kinds of controls to ascertain that the animals use a high-level rule-guided strategy to be able to attend to the appropriate target stimulus. We control the position of the animal. The animal has to head fix itself voluntarily in order to perform the task, etc. So we've done all the controls in order to make sure that mice can actually do this type of behavior. And that's really important because then we can ask questions that you cannot ask in other organisms, such as if you inactivate the prefrontal cortex of the mouse on a trial-by-trial basis, how do you actually impair the performance in this task? And therefore, what do you conclude about the causal engagement of the prefrontal cortex in goal-directed attention in this particular paradigm? And that's exactly what we did. So if we inactivate the PFC either during the queuing or the delay period of the task, the animals basically guess. It's as if we do not give them a cue at all. The behavior is random, equivalent to catch trials. If we inactivate the PFC at the moment of target delivery, the animals do the task just fine. And that level of precision in terms of perturbation is really exciting because we can tell then that the prefrontal cortex is setting up the circuit to optimally detect the target stimulus and or ignore the distractor, but not for the sensory processing itself, which makes a lot of sense, but it's hard to demonstrate in other preparations. So to make a long story short, after many, many, many studies, what we've concluded is that the way this circuit works at a macro scale is a biased competition. So it's a race between vision and audition that the PFC arbitrates. So on trials, in which the animal attends to audition, the prefrontal cortex suppresses visual transmission at the level of the sensory thalamus. And on attend to vision trials, the PFC does the opposite on the auditory pathway. And the way we know this is by recording in these different areas, observing trial specific modulation by the cue that was provided on that trial, and the elimination of this neural modulation by PFC inactivation on single trials. All right. So we also use multi-electrode recordings to be able to infer sort of neural mechanisms and look at neural signatures of this task. And again, I apologize. Actually, I'm not including a lot of raw data here because I think it's probably a lot better and easier to just give you the high level summary of these studies. And I have data in supplementals, slides if anybody's really interested. I'm happy to talk about this offline as well. But I'm summarizing here also a lot of neural signature work. We've recorded single cells. We've looked at rasters, peristimulus time histograms, made sense of what these neurons encode. But to make a long story short, what we think happens is that the cues come to the prefrontal cortex and they're maintained based on the lateral connectivity within the PFC. And we think that during learning, what happens is that there are these ensembles or coalition of neurons that become more strongly connected based on based on a specific cue that the animal has learned. And consistent with this idea, we find that the activity patterns are maintained in the PFC by bilateral connections. And this is, for example, these are two different ways to infer connectivity in the PFC that tells us that what we're looking at is we're making the right inference. These are spike time cross-correlograms, and these are connectivity filters that we get from generalized linear models when we fit simultaneously recorded neurons from the PFC. But what we think that tells us is that if neurons are encoding the same cues, they're more likely to be connected to one another and more likely to maintain the cue relevant activity patterns over time during the delay. But one of the things that we weren't sure about is how abstract are these representations in the mouse prefrontal cortex. So to take the mouse seriously as a cognitive model, one has to be able to observe things that are invariant to the low-level sensory details of the task, right? So I'll give you the intuition. Imagine that you're driving down, with your car, and you hit a traffic intersection. And if you observe a stop sign or a red traffic light, there ought to be something in your brain that encodes these very different sensory stimuli the same. Something in your brain that would trigger you hitting the brakes has to be invariant to how these sensory signals exist in the world. So what we wanted to ask is, in the mouse, is there invariance to the type of cues that we deliver in our task that tells us that the animal is really deploying attention in a cue or stimulus invariant manner? So we designed a task in which we had the mouse play two different games. Basically, the original game that I showed you, attend to vision, attend to audition, based on different past noise inputs. And then we transitioned the animal to play the same game based on different colored lights. And lo and behold, what we found in the PFC, there are neurons that can encode the rules regardless of how they were cued by these low-level sensory inputs. And that was mind-blowing for us, studying the lowly mouse, that could show something as abstract as encoding of these high-level cognitive variables in this tiny area we call the prefrontal cortex. So what we think happens is that we can think of the PFC as a recurrent network that gets inputs from, we don't exactly know where at this point. These inputs are maintained in working memory, in the PFC bilateral connections, and then they're locally decoded by neurons that encode these, that are basically, they're locally decoded by output neurons that project elsewhere and implement this idea of attend to vision and attend to audition. And what we ultimately found is that there's a hierarchy of cue to rule transformation in the PFC, and that these neurons, the attend to vision and attend to audition neurons, are actually labeled lines that go ultimately into sensory systems and suppress the distracting modality. So the mouse allows you to do these kinds of neat tricks of labeling these neurons based on hypothesized projections, and then do things like optogenetic tagging, identifying them, and then looking at their task engagement. And what we think we've identified are these output labeled lines that locally decode recurrent connectivity patterns in the from other neurons in the PFC, and project these as gain modulation signals to modulate attentional sensory gain in downstream circuits at the level of sensory thalamus. So just to conclude, and just to add one point, the intermediate step between prefrontal cortex and sensory thalamus is actually the basal ganglia, sensory components of the basal ganglia that translate that signal to sensory thalamic gain modulation. So just to conclude this part, the mouse PFC can build hierarchical representation in a task relevant manner. Sensory selection for broad stimulus features can be performed at the level of sensory thalamus, and the striatum is involved in transmitting PFC output to sensory gain control relevant for attention. Okay, so I'll quickly go through the next part and then I'll open it up for questions. So this is really, I think, the coolest part of the talk. And so we were wondering, basically, where is the PFC getting these cue inputs? And the first hypothesis that we came up with is, well, it's got to be coming from the thalamus, right? That's what V1 does. Where is it coming from? From the LGN. So where sensory input's coming from? This is the largest thalamic input to the PFC, so it's got to be from here. And we quickly realized that this was wrong, because when we suppressed the MD thalamus during the cueing period, there was minimal impact on the behavior and the physiology. Okay, but when we suppressed the MD thalamus throughout the delay period, although early neural signatures in the PFC were intact, the working memory delay period reverberant activity was gone. So what we concluded at that point is that the thalamus is not really transmitting the cue to the PFC. The cue makes it just fine. It's required for maintaining this recurrent activity that maintains the cue information over time. And that was a role for the thalamus in working memory that was, described by other people at the same time, including Carl Svoboda's lab. Okay, so then we recorded from the MD thalamus. If it's not relating the cue, then what is it doing? And it took us a few studies to actually understand what it is that it was encoding. And what we found that, in contrast to the PFC, that's encoding either the inputs and the outputs of the task, the rule, for example, the thalamus isn't showing that at all. What it's encoding is the temporal context, what game the animal is playing. And that's what I introduced in my introduction when I was saying that there is some intermediate level computation of compression that the thalamus was doing. It was really based on this initial finding, that what game the animal was playing, which was basically which cues were being played in any particular context, that's what showed up in the thalamus very strongly. So what that allowed us, and that was completely dependent on cortical input. So what we started concluding initially based on these findings is that what the MD thalamus does in order to account for these types of responses, it gets convergent input from these memory chains or memory recurrent networks in the PFC, and does a form of dimensionality reduction. It learns these types of input patterns. And when it goes back, in order to maintain these memories over time, the working memory over time, our hypothesis was, well, it must be enhancing excitability and getting these neurons to sort of like become active enough to talk to each other. And that was completely wrong, because when we activated the MD thalamus and simply asked, if you enhance excitability in the MD thalamus, what do you get in the PFC? What we found is that in contrast to sensory systems, we've done this in the LGN, looked at V1, we've done this in the MGB, looked at A1, and the effects were excitability here equals excitability here. And in this system, we did not find that. What we found is that while fast spiking or inhibitory neurons are activated by thalamic input, excitatory neurons are mostly suppressed. And that was really strange. That was unexpected. But all measures of functional connectivity between neurons in the PFC went up. So although we're suppressing baseline activity, the neurons, the inference is that the neurons are talking to each other more. You can think of it as a signal to noise type of thing. You're suppressing the overall noise of the system, but you're making the system functionally more recurrently connected. So this is not some optogenetic trick. The task does that for us. So if we look at the animals and compare the spike rates in the MD thalamus as they engage in the task compared to a baseline of just hanging out in their home cage, or during the delay period, spike rates go up by 40, 50% in the thalamus. Fast spiking neurons in the cortex go up. Regular spiking neurons, not so much. But all measures of connectivity, recurrent connectivity in the PFC, go up in a commensurate manner to the thalamus. So our conclusion so far is that the thalamus compresses these, high dimensional cortical signals and then goes back to implement two different types of functions. One is the amplification of recurrent connectivity patterns, and we think we now have a circuit mechanism for this that involves disinhibitory interneurons, and I can show data on that if anybody's interested. And another function that's broadly suppressive by contacting PV positive or fast spiking interneurons. And together, this network implements a form of balanced amplification. So when a stimulus comes in, you're not only maintaining it over time, but you're suppressing everything that's potentially competitive with it. And when the input pattern changes, you suppress the other competitive input pattern. And more recently, what we found is that those two functions are actually subsumed by two different types of thalamic neurons in the MD that have different input-output connectivity patterns and even gene expression patterns that allows us to access them independently and modulate these functions independently in the prefrontal cortex. And what we think those are important for, this inhibitory function in particular, is important for cognitive flexibility, for task switching. So what we think happens, for example, when the animal is playing two different games or switching between two different tasks, this is in the two different high pass, low pass noise versus two colored light. What we think is that there are two different configurations, two different input patterns, that when game one is on, there's a dedicated thalamic circuit that amplifies it, and then another thalamic circuit that not only suppresses one of the competing patterns, but also suppresses the other task completely, so that the local decoders, whatever is projecting outside of the PFC, can learn the recurrent activity without interference. And then when the input patterns switch completely, now you suppress the old patterns and you can learn the new patterns quickly without interference. Is there any evidence for that? Yes. This is data from animals. These are connectivity filters in neurons within the PFC at the moment of task switching. So these new input, I'm almost done. These new input patterns take about 5 to 10 trials to stabilize. These connectivity profiles, this is from generalized linear models, take about 5, this is in real animals, from 5 to 10 trials to stabilize. But when we suppress the MD thalamus on single trials, we prolong that switching latency. And when we look at the behavior, again, when this is the mouse playing task 1, switching to task 2, when we suppress the MD thalamus only during the queuing period, no effect, but the switching latency becomes longer. Okay, so I'm done. Under certain conditions, the MD thalamus does not operate as a relay. The MD thalamus exerts 2 distinct functional effects on the PFC that appear to be subsumed by 2 functional MD types. MD-dependent PFC amplification is critical for task maintenance, and MD-dependent PFC suppression is critical for task switching. And this is all the people who've done the work. Here they are, collaborators and the support. And thank you very much for your attention. I'm sorry this took longer than I intended.
Yep, no worries. Thank you very much. That was interesting. I know we have a few questions that have come in. I wonder if we want to first take a quick look at what the panel has on their mind. I've seen a few panel members join, not to put the spotlight on you, Mac, but Mac Shine has joined us from Australia. So I wonder what his thoughts are on thalamocortical interactions that I know he's very interested in. I'm going to start with an audience question first, if that's okay. Dan Lurie and Andrew Lin have a related set of questions here on how this relates, the work that you've been describing relates to interactions between the striatum and the prefrontal cortex. Dan or Andrew, if you're out there, we'll do our usual little trick. And I see a hand raised. Dan, you should be able to.
Okay, great.
Yeah.
Thank you, Mike, for that great talk. And I think this is really important work. And I think you touched on this very briefly at the beginning of your talk. I'm just curious how you think about this new work fitting into models of striatal PFC and more in general corticostriatal loops. I think, you know, the classic view of corticostriatal loops is that they go through the thalamus, and we tend to kind of ignore that part. Do you see the work that you're doing showing kind of this being part of that view, or a more direct interaction between thalamus and cortex kind of that doesn't necessarily route through the basal ganglia?
So it's a great question. So I want to highlight in the work from one of the, a very, very talented student in the lab, Rabiba Wan, who is now on the archives. I can actually, I can actually put it in the chat box. It's basically the idea is that it's not dissimilar from the idea that Matt Botvinnik has sort of suggested, this idea of a fast and slow loop within the basal ganglia thalamocortical system. So the intuition is that the thalamocortical loop is a fast loop that allows for rapid adjustment of cortical dynamics on the fly, pretty much. But it's under a corticobasal striatal sort of reinforcement learning algorithm that can select these different types of thalamocortical loop to allow for things like generalization across different tasks. And I'm just copying the link. I'll put in the chat box. This is articulated in Barbiba's work, and we're doing the theory and the simulations right now that should capture the sort of our idea of the computations. Now, what I want to also address is I probably confused everybody when I talked about the basal ganglia, because I was talking about a pathway that we've identified in the basal ganglia that has nothing to do with this idea of corticostriatal, GP or SNR pathway back to the thalamus. I was talking about a pathway in the sensory striatum that's more of an open loop type of configuration. So prefrontal input go to there, to the striatum. And then from the striatum, they go to the GPE. And from the GPE, they go to the thalamic reticular nucleus. And it's really just an open loop type of thing that adjusts the gain on sensory signals at the level of sensory thalamus. So it doesn't have to, it has nothing to do with sort of going back to the thalamus and selecting different functions at the level of thalamus. It's a different type of basal ganglia than the one that I talked about first.
Excellent. Thank you very much for that answer and the question. We have two more audience questions that I can see. One is by Shervin Safavi. Shervin, are you listening? If so, please go ahead and raise your hand. There you are, and I'll turn it over to you. There goes the mic. You're ready to go.
Hi, thanks for your great presentation. I was curious about two things. One of them is in primate literature, one of the nucleus in thalamus is pulvinar, which is involved in a lot of such tasks. I was curious if in your framework you also consider a specific role for pulvinar corresponding to mice. And the second question is, do you also think ACC is also involved in such tasks in your framework or not? And if yes, how?
So it's a great set of questions. So the first question is about the pulvinar and its role in regulating interactions across sensory, across the extrastriate cortex, for example. Is there a role for the thalamus in regulating interaction, in the medial dorsal thalamus, for example, in regulating interactions across frontal areas that would include something like the anterior cingulate? Is that am I capturing your question generally?
Yes, of course, yes. I was curious about each separately, but you actually put them both in one framework. That was even better than what I expected.
Great. Yeah, so this is on our minds, and I think so. We've been inspired by work in the primate literature about the anterior cingulate and area monitoring, and whether some of the computations that the anterior cingulate may be doing, like integrating error feedback over trials and weighing them by something like perceptual confidence, is something that can be gated by the thalamus. So we had a specific hypothesis based on findings from one of my friends and colleagues, Mehrdad Jazayeri at MIT, who's done these experiments in macaques, and under those conditions, his lab sees these error signals that are modulated by confidence that arise in the context of a hierarchical reasoning task. So we tried for, I think a couple of years to get a task like that to work in mice. It just doesn't work. So what we ended up doing, I can share my screen again, is implementing this. Oh, can you see my screen?
Yep.
Can you see this animal?
Yep.
Yeah, so this is the tapaya or tree shrew. And we're doing a lot of our experiments right now in this animal. And the reason why we're doing it is because we can still do optical perturbations in its frontal cortex. It has a primate-like visual system and a primate-like frontal system. So it has a layer 4 prefrontal cortex, whatever layer 4 in prefrontal cortex does, it has it. And then we can do something like, hierarchical reasoning. We can basically switch that. We can do a reversal in the task, for example, and then ask the animal to infer how the tasks switch based on its confidence levels. And we can see this modulation that captures what human participants and macaques do. There's no way to get something like this to work in mice, but we can now get it to work. And I guess I'll have an answer to your question in maybe 3 months or 6 months, optimistic.
Thank you very much for that question and answer. We have a couple of questions that one just came in a few seconds ago. They both address the issue of of disorders and how your work might be relevant for understanding disorders. There's Stefanos Puskas, who was first, and Morris Friedman second. Both of you might want to ask your questions. Stefanos, if you go first, I see you, and I am giving you the mic.
Yes, thank you very much. Thank you for the very interesting talk. So I was wondering, in the case of diseases with very prominent thalamic pathology, such as multiple sclerosis, what do you think, based on your findings, what can the impact be of thalamic degeneration on measures such as network controllability, but also the PFC function specifically? And I was also, given the interneurons prominent in the thalamus, I was also wondering, what is the impact on the excitation and inhibition balance that the thalamus is a gatekeeper of?
Yeah. So, so I don't know about multiple sclerosis, but I do know about schizophrenia. So I'm a psychiatrist. I see patients with psychotic disorders, and I really think about inference a lot, because one of the things that strikes me about patients with schizophrenia in particular is that they jump to conclusions that are very, very unlikely based on the things that are generally have likelihoods that are very, very small and should be discretized to zero. I think we all discretize them to zero, seem to be maintained and seem to somehow blow up over time and become real fixed beliefs that don't make a lot of sense to people who don't have these conditions. I mean, maybe under certain conditions or in certain worlds, these are actually really important things to have. But that's something that I've always found to be a feature of this illness. So what I've come to sort of think about more recently is that perhaps what's going on is that there is there's suboptimal weighing of their belief statistics over time that doesn't allow them to reach to optimal decisions about the outside world, because there's a lot of noise in the system, for example. And maybe the reason why there's a lot of noise in the system is because there's some low dimensional control somewhere, like the thalamus, for example, that's not capturing the variance of their inputs, belief inputs optimally, and therefore not able to exert. Either the variance is not being optimally represented or captured, or the control based on that representation is suboptimal. It's not entirely clear. But that allowed us to start thinking about studying decision-making under uncertainty as a proxy for the decision process in psychosis. Okay, so I'll share my screen again. Is that okay, Olaf?
For a minute is fine. Yeah, go ahead.
So, there are these findings in humans with human fMRI, is that the medial dorsal thalamus, the structure that I told you about, is particularly sensitive to uncertainty in decision making. This is a categorization task. This is a multi-attribute attentional task, and in a lot of these, this is modeling from Xiaojing Wang's group in the pulvinar, which is very similar. Which, again, to one of the previous questions, again, capturing these types of statistics, variance or the inverse of variance, is sort of implemented in these models as a convergence operation that I sort of pointed out early in my talk. So we became interested in this, and we ended up implementing a decision-making task in mice that's a probability. So that basically takes the attentional task and makes the inputs, parametrizes the uncertainty of the inputs. And to make a long story short, I'll show you one piece of data. When we suppress the MD thalamus in that decision-making task, mice become much more sensitive to input uncertainty. Okay, so they become less able to do the task when the uncertainty rises. Okay, and we do see signals in the MD thalamus that are low dimensional and are about the variance of the input. Now we have, I'll just fast forward, I'll just make my point. We have an ongoing collaboration at Vanderbilt with Anna Huang and Neil Woodward, who study who use DTI to look at structural and functional connectivity in this frontal thalamocortical network, and they've described deficit schizophrenia. So we've implemented this task in humans, and what we find is that schizophrenia patients have a deficit that's very similar to mice with MD inactivation, optogenetically. So that's the closest I can get to trying to answer. It's a long-winded way of saying we decompose the problem that you posed into, this really contrived tasks that hopefully approximate the sort of inference and decision-making process at this most elemental level. Whether we're right or not, or these parallels are the right parallels, I don't know, but that's what we're doing.
Thank you very much. I hope that answered your question, Stefanos. I looped in Morris because
Yes.
he had a question related, I think, if I'm
Yes.
reading your question correctly. So over to you, Morris.
Yes, great talk. Alcoholic Korsakoff syndrome is actually a human model of dorsal medial nucleus of the thalamus lesions. So I guess the question is whether anybody's tested patients with this disorder using your test, and if not, would that be worthwhile to
It's a
see if
great
it
question.
translates into humans?
It's a great question. Okay, so the short answer is I don't know. The longer answer is I'm not sure. So the medial dorsal nucleus is a very large one. It has a parvocellular component, a magnocellular component, a lateral component, a medial component. The Korsakoff syndrome is associated with degeneration of the medial component of the medial dorsal nucleus, which is connected to the mammillary bodies and is connected to the medial portions of the frontal cortex. What I'm talking about here is specific to the lateral executive portion of the MD thalamus. So I don't know how much of the principles that I talked about reconfiguration, applies to this other circuit that's impacted in Korsakoff syndrome.
Okay, so it sounds like it'd be a model to test that other circuit.
Yes, which I know nothing about.
Okay. Sounds interesting. Perhaps the final question from the audience coming from Mike Schmidt. If you're out there and ready to raise your hand, here we go. Over to you, Mike.
Hey, a lot of this I think is due to my own naivety in understanding this, but I do fMRI work and I'm trying to think how I would integrate what I just learned from you into emotion regulation and executive control doing human fMRI at the much lower resolution in both space and time. And both of your last answers actually gave me great places to go and look at that question. But if you have anything you would share on maybe how to use your findings in interpreting human work at that resolution where you're non-invasive and you can only look at things in a certain way.
Yeah, that's a great question. I mean, I think one prediction that I would imagine this work would do is that if, one was doing, multi-voxel pattern decoding, for example, that what I imagine the prediction here is that looking at frontal cortex, these patterns will be a lot, higher dimensional than the ones in the thalamus. And I think one of the values of having these compressed representations in the thalamus is that, for cortical-cortical interactions, it's far easier to have these compressed representations just being sort of projected from one area to the next, rather than having the high dimensional sort of representation in one area be mapped onto another area. It's just a sort of from a control perspective, it's probably a lot more efficient to do this through the thalamus. So the prediction would be is that if you're decoding these multivoxel activity patterns, that you'll find them to be compressed in the thalamus, and then you know, I think right now people are doing things like focal ultrasound to perturb circuits deep in the brain. So this might be, one might be able to target the thalamus in particular phases of a task to test the model predictions as to, you know, how inference or emotional regulation will vary as a function of thalamic perturbation.
Thank you
Very
so much for your
interesting.
thoughts.
Yeah, very interesting. Thank you, Mike, for the question, and thank you both, Mikes, for the question and for the answer. Thank you. Are there any other questions from the audience or the panel?
Yeah, I've got a question, Oleg.
Mac, there you are. Great. Good morning.
Hi, Mike. Thanks for the great talk.
Thanks.
It's nice to finally be able to join one of these. They've been on at like 1 a.m. Australia time. So Mike, I'll take the bait a little bit on your nice sort of idea about some of these thalamic nuclei coming up and inhibiting areas and then projecting to other areas and sort of controlling kind of cross-regional gain. And you mentioned that they have genetic markers. Are you thinking about things like the kind of parvalbumin calbindin story that Ted Jones talked about, or are these both matrix type thalami that have different kinds of projections? I'm really curious.
So no, they're not calbindin positive. I mean, I don't know if they are. I don't think they are. I think we've tried this calbindin thing. I think what we've been able to do is to, you know, I can show you one piece of data. Well, I can just tell you because, you know, I don't want to slow this down. What we've done is based on the circuit hypothesis that one in the lateral MD, at least in this very small part of the MD thalamus, we have two functional cell types. One that seems to, you know, amplify recurrent connectivity pattern. One that suppresses activity generally. So we had the hypothesis that, well, maybe they're implementing that by projecting to, you know, generic inhibitory neurons PV positive, and disinhibitory neurons VIP positive. So we started by labeling these two different populations from the cortex with rabies, and then tracing back the thalamic afferents from the MD thalamus. And they just simply labeled different populations, spatially segregated populations in the MD thalamus, and they have different genetic profile. I don't think that calbindin sort of co-varies across these profiles. Frankly, I don't know if matrix and core really apply, are the most helpful types of nomenclature for these types of differences. I think probably we need a new way of being able to identify these things. Because
yeah, I hadn't appreciated that you'd started in the cortex with a particular cell type and then tracked back, which I think is actually, you're quite right, is something's missing from the nomenclature at that level of specificity. We have the notion of first order and higher order from Murray Sherman and colleagues, but really that's sort of developed in the LGN and makes a lot of sense when you think about propagating a really precise signal. Yeah. If you're talking about context-dependent modulation of, you know, descending cortical circuits, it's a much more nebulous concept. So I really like that idea, and would encourage you to maybe think about SST neurons as well and complete that nice little tri-circuit that Carol Svoboda has been showing.
Yeah, we've done that. We've done that as well. It's just that the segregation of function in that loop does not seem to care about SST.
VIP, PV, got it. Cool. Yeah, awesome. That's great. Thanks for the answer.
Yeah, no problem.
Thank you both for that discussion.